Hey everyone, and welcome to This Explainer. Today, we're actually tackling a really fascinating paradox right at the heart of modern biology. You see, over the last 20 years or so, our ability to sequence DNA has just absolutely skyrocketed. It's actually outpaced Moore's law for a long time. We're now routinely cranking out genomic data on a petabyte scale. But here's the catch. All that raw biological data, it's basically useless on its own. The real bottleneck in genomics today is no longer the sequencing part, it's the computational analysis. We literally have the complete chemical blueprints of life sitting right in front of us, but our human brains just cannot read it manually. To actually extract meaning from these massive datasets, we need an entirely different kind of digital infrastructure to make sense of it all.
Which brings us to a huge question. Can computers actually understand DNA? It's a pretty wild computational challenge when you really think about it. I mean, we're not just looking at a simple linear book here. The human genome is made up of over 3 billion base pairs. And when scientists sequence a genome, they don't just get a neatly formatted document delivered to them. No way. They get this massive, completely fragmented data set that has to be computationally reassembled, aligned, and annotated. It's like a giant puzzle. To actually unlock the biological mechanisms driving health, disease, and even evolution, we absolutely have to rely on these digital frameworks acting as translators, processing this immense scale of information entirely without human intervention.
And the core reason we need so much massive computing power? Well, it really just comes down to high-throughput sequencing technology and sheer overwhelming volume. Modern sequencing machines, like the Illumina NovaSeq, can generate terabytes of raw data in literally just a couple of days. But here's the crazy part. They don't read your genome end to end. Instead, they blast it into hundreds of millions of tiny fragments, often just 150 base pairs long. So the computational challenge is taking the absolute avalanche of disorganized pieces and mapping them all back onto a reference genome. It is a massive algorithmic puzzle. Just one single human genome can eat up about 100 gigabytes of storage in its raw computational form. Multiply that by population-level studies involving tens of thousands of individuals, and wow, you are suddenly dealing with exabytes of biological data that must be structured, parsed, and analyzed.
Section 1. Organizing the genetic code. Okay, let's dive into this and see how scientists solve this massive data bottleneck. The discipline that comes to the rescue here is called bioinformatics. Simply put, bioinformatics is the use of computers and software tools to process, visualize, and transform raw biological sequencing data into meaningful insights. It acts as the vital translator for the genetic code. But it's way more sophisticated than just dumping biology into a spreadsheet. Bioinformatics is the architectural bridge between raw sequencing outputs, like massive FASTQ files, and actionable scientific discovery. So the really crucial point here is that these software tools deploy advanced statistical modeling and complex algorithms to clean, align, and process the data. They filter out sequencing errors, they piece together those overlapping fragments using things called de Bruijn graphs, and they visualize complex molecular interactions. Without this highly specialized computational framework, the entire field of modern molecular biology would effectively grind to a halt under the weight of its own data.
Now, what's really interesting about this slide is how this computational pipeline operates in practice, because it forms the very foundation of bioinformatics. It follows a highly structured, logical progression. First up, storing the DNA data requires some seriously advanced cloud computing architectures and aggressive data compression algorithms just to keep these petabytes of data accessible and secure. Second, comparing sequences relies on dynamic programming and heuristic algorithms to rapidly find genetic matches across billions of letters. Third, once it's all aligned, the software identifies mutations. It pinpoints single nucleotide polymorphisms that deviate from a baseline, while very carefully filtering out any false positives caused by machine errors. And finally, sophisticated statistical tools take over to detect broader biological patterns. They scan across thousands of genomes to figure out exactly which genetic variations correlate with certain traits or diseases. It's essentially a brilliant funnel that distills raw noise into high-fidelity scientific signals.
Section 2. Real healthcare applications. Now let's pivot from the abstract data side of things to the concrete human stakes and emphasize exactly why this matters.
This brilliantly illustrates the real-world impact. Bioinformatics is literally the hidden engine actively powering modern healthcare breakthroughs. Let's look at cancer research. Tumors are now routinely sequenced to identify their specific genomic drivers, and bioinformatics pipelines analyze that data to match patients with highly targeted therapies, totally avoiding the trial and error of broad-spectrum chemotherapy. For genetic testing, this computational power is what makes personalized medicine a reality. I'm talking specifically about pharmacogenomics, allowing clinicians to predict if a drug will be life-saving or toxic based solely on your unique metabolic pathways. And in drug discovery, computational models can rapidly screen millions of potential molecular compounds against a genetic target, compressing years of tedious lab work into mere weeks. The rapid development of mRNA vaccines, for instance, relied heavily on bioinformatic mapping to optimize the genetic sequence of the viral spike protein before a single physical vial was ever manufactured. It's a total game changer.
Section 3, AI and Biology's Future. So, let's introduce the next frontier and look ahead at the increasing role of artificial intelligence in biology. Computational biology is currently undergoing a massive evolution. Artificial intelligence and deep learning models are completely revolutionizing how we handle this data. We are moving way beyond traditional algorithms straight into neural networks that are capable of predicting highly complex biological behaviors. A perfect example of this is AlphaFold's recent breakthrough in predicting 3D protein structures directly from 1D genetic sequences, a problem that literally stumped scientists for 50 years. AI just excels at finding subtle, nonlinear correlations in multi-omic datasets that human programmers wouldn't even know to look for. But, and this is absolutely crucial, this is an incredible symbiosis, not a replacement. Computers aren't replacing biologists. The AI generates high-probability hypotheses and does the heavy analytical inference, sure, but human scientists still take those insights into the wet lab to carefully validate biological viability and clinical safety. Basically, the algorithms point the compass, but scientists still steer the ship.
So, we've seen how these sophisticated computational pipelines manage the terabytes of raw fragments generated by high-throughput sequencing. From aligning billions of base pairs to enabling hyper-targeted cancer therapies, the digitization of genomics has fundamentally rewritten the rules of medicine. And you know what? The future is only going to get faster and more precise as deep learning models unlock even deeper patterns hidden inside our cells. It turns out, the ultimate code isn't binary, it's biological. In fact, scientists are currently experimenting with using synthetic DNA as a high-density medium for storing digital data itself. How wild is that? So, I want to leave you with this final provocative thought. If biology is now data, what will the next big computation cure? Thanks so much for joining me on this explainer, and keep questioning the data around you.